Hey, so today we're going to talk about high level respiration. It's one of those topics that takes a while for that eureka moment to come about, where it all kind of clicks into place. So I like to always think about the big picture first of all, and then like zoom in on the details when I talk about it with my students. So I've got like the option to change this to dark as well. I think some people prefer this. And actually on my site now I've built out where you can change the color palette too, just to hopefully make sure it's a bit more user friendly. If it's still not quite right for you, let me know, and maybe I can just add a simple black and white version for everything too. It's not a bad idea. So when we think about cellular respiration, where does it all take place? So glycolysis is happening in the cytoplasm. I've given a basic overview of the SL content in another video. We're going to focus on the high level stuff here. So we already know about glycolysis. The end product of glucose goes in, it's broken down. So it's what we call a breakdancing catabolic process. So catabolism. So glycolysis is going to produce 2-pyruvate. You want to know the steps of glycolysis. So first up, we have phosphorylation. So ATP adds phosphate to the glucose. It causes a destabilization because if you add something to a molecule or take something away, it causes a structural change. And that can cause instability or also loss of function. So here we've got instability. That leads to lysis. So we have phosphorylation, lysis, hence the lysis part of the name. So glycolysis. So the glyco is the carbohydrate aspect. Lysis means splitting. We use that when we talk about cells bursting. Like a red blood cell put in a distilled water solution is going to take in too much water and burst. It's got no cell wall to stop that from happening. So we have phosphorylation, lysis. We've now got 2,3 carbon compounds. And then we have some oxidation. So oxidation is loss, reduction is gain. So the molecule itself is oxidized. That means it has hydrogen stripped away. And then we have some substrate level phosphorylation. This hydrogen being stripped away is really important. So the whole process of cellular respiration, I always think is just building up to producing, and I think it because it's true, is building up to produce a proton gradient. And this is where the electron transport chain comes in, and we're able to produce ATP. So what I mean by proton gradient is a hydrogen ion. So hydrogen is stripped away from this 3-carbon molecule in glycolysis, and a hydrogen ion is just the proton. So we use this term interchangeably, these terms. So we refer to it as a hydrogen ion or a proton. And NAD is our carrier. So NAD is reduced, reduction is gain. So there's a mnemonic I'm using here, oil rig. Oxidation is loss, reduction is gain. And it's to do with the gain of electrons or the gain of hydrogen. Oxygen itself, like if something is oxidized, oxygen is added. Or if it's reduced, oxygen is taken away. But when we're talking about hydrogen, ions, and electrons, oxidation is loss, reduction is gain. And you can see every step along the way here is trying to build up and create hydrogen ions that will get taken to the electron transport chain. So these guys here also go down, this arrow should extended, and ATP, a lot of ATP will be produced. So glycolysis is referred to as like the common pathway or fermentation reaction, and it's part of anaerobic respiration too. So it's something that is a precursor to the rest of aerobic respiration, and the other stages will occur if there's oxygen present. Because oxygen, let's skip down here, is super important. And this is our electron transport chain. Oxygen is super important. It's what we refer to as the terminal electron acceptor or the final electron acceptor. And without oxygen, the whole process just like ceases to occur. So if we drop off some NADH, you can see ATP synthase here. Some hydrogen ions has gone into our intermembrane space, And all that's happening, and I'm going to explain the rest of the details in a little bit, all that's happening is hydrogen ions are moving down their concentration gradient. And they're moving through an enzyme called ATP synthase, and it does what it says on the tin. It produces ATP. So it takes ADP and adds a phosphate. So you go from adenosine diphosphate to adenosine triphosphate. Of course, that is a nucleic acid. ATP is a nucleic acid. So cho cho chom chomp. It's got carbon, hydrogen, oxygen, nitrogen, phosphate. And it's only able to do that when the electrons can flow through here. When the electrons can flow, then hydrogen ions are moved in. So the NAD drops off electrons, it drops off hydrogen, and that hydrogen ion gets pumped in. Now we say that FADH produces the equivalent of 2/3 of the amount of ATP when, like, relative to each other, if you had equal amounts of them. And that's because it's dropped off here at the second protein carrier, and it goes through 2 pumps. So it only moves through 2 pumps. You can see only moving hydrogen ions through 2 pumps. So, let's go through two, so 2 out of 3. But we don't have equal amounts of it. But just sometimes you might get asked a question that has that kind of comparison about the amount of energy that they're bringing, and it's the electrons only going to move through 2 pumps, so it's only able to produce 2/3 relative amount of ATP. So let's look at some of the details I skipped over. FADH2 is produced down here in the Krebs cycle, but before that, we've got the link reaction. So the link reaction and the Krebs cycle, they're occurring in the matrix. So this is a mitochondria, not the best mitochondria. I mean, we wanted to keep these labels in here, so the cristae are not as extensive. There isn't a barrier here, but the cristae are there to increase the surface area to volume ratio. Now, glycolysis occurs in the cytoplasm, and in the presence of oxygen, glycolysis, the pyruvate end products is 2 pyruvate produced and a net gain of 2 ATP. 4 ATP are actually produced, but 2 were needed in order for that phosphorylation at the beginning, because you have phosphorylation, lysis, oxidation, and substrate level ATP production. And then that pyruvate goes and is now available for the link reaction. We've referred to the link reaction as oxidative decarboxylation. So click on it down here. So here is our decarboxylation. So decarboxylation, losing carbon dioxide. As simple as that. And the main thing that we're getting here is a little bit of NADH, which is going to carry some hydrogen and make it available for the rest of the reactions. Now, the Krebs cycle comes after that. And the Krebs cycle is also going to produce some CO2 and is also has some oxidation, is also oxidative decarboxylation. So we've got substrate level ATP production too, which we don't have anywhere other apart from glycolysis. The link reaction is the one that misses out here. No ATP is produced there. But when we think about the Krebs cycle, it's a cyclical event. So acetyl is produced. It's a 2-carbon compound at the end of the link reaction, and coenzyme A helps make that available. So you have acetyl coenzyme A, or acetyl-CoA. And you can think of CoA as a molecule that's just helping acetyl get to where it needs to go. And it's going to be bonded to a 4-carbon compound. Now we actually have to remember the first 4-carbon the 4-carbon compound at the end of the Krebs cycle that then gets joined with the 2-carbon compound. The way I remember it is oxalacetate is our 4-carbon compound, and oxalacetate, I remember it because I just think of a picture of an ox. And then citrate is our 6-carbon compound, and this is often referred to as the citric acid cycle. Okay, so at the end of the citric acid cycle, and that's the way we remember, citrate is our first 6-carbon compound, oxalacetate is the 4-carbon compound at the end. No idea why IB have suddenly decided that we need to remember the name of these two. It's just a bit more rote learning for you, but all of these key terms, they're all in Cramly, my free study app with flashcards. So I really recommend downloading that, available in the Play Store. If you're an Apple phone, you can just save it to home, and then you've got it as an app that has some, has a database, so it'll track your learning and has an adaptive algorithm. So yeah, I really recommend checking that out. It's really helped level up some of my students. One guy went from a 4 to a 7 because he really engaged in the process. But if we move back to this, the whole purpose is about creating a proton gradient. And that whole purpose of a proton gradient, we know that if things diffuse, they move from an area of high concentration to low concentration. Another way that we get to this high concentration gradient is this intermembrane space. This is where the hydrogen ions are moved to. So you have the Krebs cycle, the link reaction occurring in the matrix, and then we have the hydrogen ions accumulating in the cristae, in the intermembrane space. The cristae is part of the intermembrane space, but the word refers to these like finger-like protrusions. And we know that that's going to increase the surface area to volume ratio, which means it's easier to maintain a high concentration, and it's also easier to get there. Now, oxygen acts as our terminal electron acceptor, and the role is twofold. It helps everything flow. Without it, the electrons won't flow, but it also mops up hydrogen ions that are flowing through here and helps maintain the concentration gradient. So it's one of these topics, respiration, where you're going to have to hear a few explanations, you're going to have to read a few texts, but it's really satisfying when you have that eureka moment, because ultimately it's just some simple ideas that we're feeding into each other. The idea of diffusion, things moving from an area of high to low concentration gradients. You've got enzymes in there. These are all enzymatic control reactions. And if you go on to do medicine, you're going to have to know the name of all the different substrates and enzymes involved in this process. Good luck to you. I do not know them. But you got to sort the wheat from the chaff somehow. And let's just finish up by having a look at a few questions. So there's lots more little facts on here for you. I don't, no benefit for me reading through. But if we put it all together in a mind map, then this is just some of the key takeaways. Glucose is split by glycolysis. This feeds into the Krebs cycle. What's missing here? Also, the link reaction. So a little bit of simplification in this mind map. But the idea of all of this is to create and load NAD and FAD. Most of it comes from the Krebs cycle. And it's all about building a proton gradient, because without a proton gradient, we don't get ATP produced. Now, as a gap fill for you, a little bit of fun, and also a few questions at the end. So let's do a couple together. So where in a eukaryotic cell does glycolysis take place? Cytoplasm. What is the net yield of glycolysis per molecule of glucose? Boom, it's wrong. Why 2 ATP? Because 2 have to be used in that initial phosphorylation. So phosphorylation is adding phosphate, and that causes the destabilization. So 4 is actually produced, but 2 were used. So we have a net gain of 2. Right, there's another 7 there for you, and I have another practice test I'll link. Feel free to drop a comment. I read all of them. I'm really interested in making these videos as useful for as many students as possible. And if you've got this far, thanks for listening, and ciao for now.